This is part 3 of muscular tissue, and we're going to be discussing muscle contraction. So we're going to talk about the sliding filament mechanism, and this is a model that describes the process of muscle contraction at the molecular level. Muscle contraction occurs because the myosin heads, they attach to and essentially walk along the thin filaments at both ends of the sarcomere, progressively pulling those thin filaments towards the midline. As a result, the thin filaments slide inward and then meet at the center of the sarcomere. The structural proteins, they pretty much hold everything together and they allow the contraction to propagate through the entire muscle. So the thin filaments slide together during each contraction, and they can even move so far inward that their ends overlap. So here you have a thin filament here and one here, and they will actually move toward each other, moving these Z discs closer to one another. And actually here, you can actually see that they overlap in when you have a muscle that is contracted as maximally as possible. So as you have the thin filaments moving inward, you have the I band and the H zone, which will actually narrow and get smaller and eventually disappear altogether. As you can see in this picture, they disappear altogether when the muscle is maximally contracted. The width of the A band, however, see that right here, and also the individual lengths of the thick and thin filaments, this remains unchanged. They don't change their length at all. They actually just overlap one another. Now remember, the thin filaments attach to the Z disc on one side. The Z disc has attachments of thin filaments on both sides of it. When the thin filaments slide inward, the Z discs come closer together, like I said before, and the sarcomere itself shortens. So shortening of the sarcomere actually causes the shortening of the entire muscle fiber, and then in turn, this leads to the shortening of the entire muscle itself. So we're going to actually talk about the specific steps of the contraction cycle. So in these diagrams, you're going to see the thin filament here and the thick filament here. So once we start a contraction, the sarcoplasmic reticulum releases calcium ions into the sarcoplasm. So here we have these calcium ions here. You can actually see them on the diagram here because they bind to troponin. So here are those calcium ions, and they bind to troponin, which remember is this blue molecule here. And once calcium binds to troponin, the troponin moves tropomyosin away from the myosin binding sites on actin. Remember, tropomyosin is going to be this orange rope-like molecule, and then we're actually going to have those visible binding sites on actin where the myosin heads can bind to. And once those binding sites are free, the contraction cycle can actually begin. And a contraction cycle is this repeating sequence of events that causes the filaments to slide. And there's going to be 4 specific steps in this contraction cycle that we will go over. So the first step here is going to be ATP hydrolysis. So the myosin heads actually have this ATP binding site and also an ATPase. And an ATPase is an enzyme that hydrolyzes ATP and converts it into ADP and 1-phosphate group. So this hydrolysis reaction actually reorients and energizes the myosin head. And after that happens, we move into step number 2. And this step, we're going to have attachment of myosin to actin to form cross bridges. So this is going to be the cross bridge step. So this energized myosin head attaches to the myosin binding site on actin, and it releases that previously hydrolyzed phosphate group. So you can see that right here. It is released into the sarcoplasm to be used by other reactions, by other things. We don't really need to know much about that, but that's okay. It's just released into the sarcoplasm. And when the myosin head attaches to actin during contraction, they're actually referred to as a cross bridge. So this here is a cross bridge. And that brings us to step 3, which is the power stroke step. So after the cross bridges form, the power stroke occurs. Now during a power stroke, the site on the cross bridge where ADP is still bound opens up. And as a result, the cross bridge rotates and releases ATP. So that kind of rotation or that movement of the myosin head actually releases ADP. And the cross bridge generates force as it rotates toward the center of the sarcomere, sliding the thin filament past the thick filament toward the M line. So we're actually going to be pulling that thin filament this way, those myosin heads. So they kind of bend a little bit like this, as you can see, and they pull that thin filament toward, they pull the thin filament toward the M line. And then we get to step 4, and this is going to be the detachment step. And this is the detachment of myosin from actin. So at the end of a power stroke, the cross bridge remains pretty firmly attached. That is until myosin binds to another molecule of ATP. So as ATP binds to the ATP binding site on the myosin head right here, the myosin head will then actually detach from actin. And this contraction cycle repeats as myosin ATPase hydrolyzes the newly bound molecule of ATP. And then this contraction cycle will continue as long as ATP is available. And remember, calcium levels are also have to be pretty high near the thin filament as well, so that troponin can actually continue to move tropomyosin from the binding sites on actin. Because if calcium isn't available, those sites are all covered and we can't have any contraction cycle. The cross bridges keep rotating back and forth with each power stroke, pulling those thin filaments toward the midline each step. Here is a playable animation, so be sure to click on this link here on your slide so that you can go and watch that animation of the contraction cycle and actually see how it works a bit better. Okay, so an increase in calcium concentration in the sarcoplasm starts the muscle contraction and a decrease stops that. We know that because when we have an increase in calcium, it binds to troponin and we move that tropomyosin away from the myosin binding sites on actin and we can start the contraction cycle. So without that calcium, we're going to not be able to remove that tropomyosin from the binding sites. When a muscle fiber is relaxed, the concentration of calcium in its sarcoplasm is very low. However, we do have this huge amount of calcium that's stored inside the sarcoplasmic reticulum. So as you can see in here, it's a lot stored, but out here in the sarcoplasm, it's going to be very low when the muscle is actually relaxed. So as a muscle action propagates along the sarcolemma into the T-tubules, you can see here we have this action potential as denoted by these red arrows here. This is going to be the electricity or the action potential propagating along the sarcolemma and then here into the T-tubules. it actually causes the calcium release channels, these little black arrow things, it actually allows them to open and allows the calcium within that sarcoplasmic reticulum to be released. The calcium will then flow out of the SR into the sarcoplasm around the thick and thin filaments. You see the calcium will kind of surround everything there. So as a result, we have this calcium concentration in the sarcoplasm rises like tenfold or even more around the thick and thin filaments. Remember, this released calcium will combine with troponin, causing it to change shape. And this conformational change moves that tropomyosin away from the myosin binding sites on actin. It's actually written out right here. Uncovers that myosin binding sites on actin. So once those binding sites are free, the myosin heads can bind to them to form a cross bridge, and that's when the contraction cycle begins. These events leading up to the contraction cycle are collectively known as the excitation-contraction coupling. This is because these are the steps that connect excitation, which is a muscle action potential propagating along the sarcolemma and the T-tubules. And it connects it with contraction, which is the sliding of the filaments. Oops. So because we're going to connect that action potential with the actual contraction, it's called excitation-contraction coupling. So this figure here shows the length-tension relationship for skeletal muscle, and this indicates how the forcefulness of a muscle contraction depends on the length of the sarcomeres within a muscle before a contraction begins, so prior to the contraction. So the resting sarcomere length, this is the most important thing when we're talking about the length-tension relationship, or how forceful a muscle can contract, is really dependent on the resting sarcomere length. So at a sarcomere length at about 2 to 2.4 micrometers, which is very close to the resting length in most muscles. This zone of overlap in each of the sarcomeres is optimal, and it's at this overlap that you're going to have the muscle fiber that can develop maximum tension. So you're going to see that here at the top of the graph here. Notice that the maximum tension, or 100%, this occurs when the zone of overlap between a thick and thin filament extends from the edge of the H zone to the end of a thick filament. So you can see that right here. So outside that overlap, you could even have more overlap to the left here, or less overlap. You're going to have, the tension is going to be a lot lower than this optimal overlap here. So you can see here we have a very steep drop when you have a lot more overlap, and then a little bit less of a steep drop, but still a drop nonetheless when there's less overlap between the thick and thin filaments. So here we have this optimal resting muscle length, this optimal resting sarcomere length. And that's going to be the end of part 3.